So Clementine is with us. So she said, do I need my laptop? But Hang is started the recording already. So
I might still join on my laptop so that I have, my files.
Okay.
If I need to look at anything.
Hi, everybody.
Share, because then it's simple.
So University of Sydney is chairing. So I just asked Clementine if she chairs or if Hang chairs.
Oh, I see.
Whom do you mind sharing off my, the agenda that I put up the other day? Or is that?
Yeah, I'll just get that one. Yep, sure, no worries. Okay, hi everyone. I think is that everybody? Are we waiting for someone? Does anyone know?
No, but since we're starting, I just want to introduce Dunya. She made this morning also her call sign for GitHub, and she started her PhD a week ago. So she will be the new PhD student from Erasmus MC working on the MycetOS project. In a month, so next meeting, we also have the postdoc here with us, but he will start next month.
Yeah. Awesome.
Very welcome.
Welcome to the group. All right. With that, the first few items are on the agenda will be for screening updates. So, the master list now includes a column that indicate, I assume that will be the date of arrival at Erasmus MC. And also there is one questions on whether what the different tabs on the master list are for. Is that when do you want to explain?
Yes, so the master list now not only has an arrival date at Erasmus MC, but for those of you are shipping compounds, it's also good to put in when you ship those, simply so that we know when they were shipped in case they didn't arrive at a certain amount of time.
Sorry, because I've put that there. I wanted to ask on the master list, there are some tabs that obviously we always use the MycetOS compounds, the first one, and then there are some tabs down the bottom, and I think one of them is out of date that says breaking good. I just need to check with Kimberly from Australia about that one. But then some of these other ones like MycetOS in vivo and in vivo potency.
Yeah, we didn't put them on. The only tabs we did was the MycetOS and the MMV. We made the MMV a separate tab because that was not compounds created by us, but simply compounds we used in the MycetOS program to be able to identify lead compounds. And since there's different data entries in the MMV, so that's why I decided to keep that one separate. And with the kinase library, that was a little bit the same. So we had different information than in our normal but still it's all the information we have. So I made a separate tab of that. But for the rest, I didn't make any of those extra tabs. And I think the EpiCam was the one we made in the beginning when we asked people, because that's, I think, was the complete EpiCam library. And then we asked people to think with us which of the EpiCam compounds we should test. So that's therefore also a separate tab, but I don't think the rest of the tabs, especially myas in vivo, that's also in the main file. So why there should be a duplicate tab, I don't know.
I was wondering if maybe it had been put there before it was moved over, but
no, it was already, it's always
Okay.
been in the same file.
So if anyone knows who put them there, maybe we can check with them and then delete it. Do you think it would have been Ma who?
No,
Did that
because
in
this is
vivo?
prior to more.
Okay.
I think one way would be we can check the version history. That should give some information. I think one or two of that might be me at some point. I will probably go and have a look, and I'll see how that goes. Yeah, I think if you check version, or if I will go and check the version history, it might just say, oh, this person with who may or may not be anonymous had created this tab. I will see.
Yeah, I mean, obviously it's not crucial to do it immediately, but it just seems silly to have tabs that are pointless repetition.
Yeah, and when I update the in vivo data, I only update it in the master file. So if it's an in vivo data for the MMV compounds, it's in the MMV compound list. If it's myos, I only update it in the myos. I don't update it in the other tabs where it says in vivo, because that seems a little bit of a waste of time to update it in 3 different databases.
Yeah, I think the myos in vivo specifically was created at one point where I think we just have a thesis or anything that just required a particular group. So I think that was created for that purpose. I think I might have created that one. Not sure about the rest of it.
Yeah, it'd be great to get rid of the duplicates if they are duplicates. And I guess if you need to create a new list temporarily for a bit of a project, you can always put a README or something in the first cell, just so it's clear when it can be removed,
So maybe an action point could be that each of us states after, in this minutes or be underneath the agenda, which tabs are essential and are still in use. If all the core members do that, then we know which tabs are never used.
Sounds good. Okay, next item. I'll getting back to the screening update in a bit, but there's one action for Alference to send more compounds for rescreening. Is there an update for that one? So Mao 626, 627, 628, and correction 137.
We didn't receive them yet, and I don't think they were shipped.
The last thing that I heard from Laurie, I think she might be unclear because she said she didn't have 5 milligrams of each of them, and she asked if she could send less, and no one's given her an answer. So she might be waiting to find out. And I didn't know how to answer her, so.
Yes, I will put that, she can send less.
Okay, cool.
I think from memory, it's about 2 to 3 milligrams is enough for duplicate testing. Is that correct?
Yeah, but in vivo we use a little bit more.
Okay.
Because that was also the issue. We run a few vivo tests with those and now they're gone.
I see. All right. For screening, the last one is mass update if there is any new results.
No, if I checked, I think everything Ma screened this year is in the list.
Okay.
Ma moved to China, so she couldn't attend today, but she's trying to attend at least the afternoon sessions, the Dutch afternoon sessions, but the morning sessions, it's now the even month, so the odd months she cannot make, but she will try to join in as much as possible and in the even months.
Yeah. Okay. That makes sense. So there will be so all of the updates for results from pharma have been provided so that we can.
Yes, so the only compounds we didn't screen yet are those from Peter because they arrived last week. And that will be probably Clementine who screens them, and otherwise Dunya will pick up from here.
Yeah. Okay. Okay, next is series 1 phenomena, and there are 3 action items. So the first one is adding structures for the I think there was 2 untested compound that was obtained from TCG and screen. Has this been assigned to anybody, or is this a fresh item?
I think it was assigned to someone last time, but it doesn't seem to be written down. I couldn't find who it was. That's why I put it in the agenda again. And the same, I think, with the next one about the out-of-date page. I think the plan was for somebody to put it all together, but I can't see who that was supposed to be.
The second one, I think I can provide an answer. I just haven't prepared for it yet, but some of these look similar to some of my stuff. I can go back and have a look at this and give an answer. I just haven't got a chance to do it yet. So maybe just assign part of that to me. I think some of that, say about 50% of that looks like my stuff. And I think some of the rest of the stuff is Dimitri's compound. So for the second item, it looks like it's me and Dimitri. So I think we can probably provide some sort of update for that, and that can be archived. But I personally won't be making any more from that set. So I'll give an update, and I think that can be archived / moved to future projects. And last item is assigned to Matt to capture these desirable compounds for, I think that will be our manuscript. Is that right, Matt?
Yeah, it's still on me. Sorry, I haven't managed to do it. Just got back from holiday yesterday, and it's been a whirlwind. So I will do it. Leave it with me.
Okay, sense. So paper updates. Well, I think there won't be any updates for the time being. I assume so. I think we can just leave that for a bit more time.
Yeah, I've got updates. I think I've got the required things from everyone,
Okay.
and I've just got to synthesize the last version, and then we get it in. But we're looking in good shape.
Okay, sounds good. Have we chosen a journal for submission? I don't think I've attended a meeting if we've discussed that.
Yeah, I think it's what is it? ACS, infectious diseases, I think, or Medcam. I forget which one it is. One of the ACS journals.
Okay. Okay, modeling updates. Next one has been archived here, which is okay. So it's been archived in protein modeling, and there is one question to would it be better to move the modeling update information to the Phenomals page or just leave it?
Yeah. In general, I didn't know whether we wanted to archive things centrally or in specific repositories. If it's, like this is modeling for the Phenomals, but it might be easier for people to find it if it's more central. So if anyone has thoughts on that.
I think modeling for the Phenomals didn't work out, but it would be for any of the other series when we have a good target or we have a sense of the target. It would be nice to do modeling anyway.
Yeah, so I started trying to, make the initial repository a little bit clearer. I thought maybe monthly Zoom meetings, the wiki was the best place to be putting this like archived work because then it's in one place. But yeah, obviously we all need to know that it's there and to tell people that it's there and so forth.
Yeah.
We've got that other repository, the what other molecules to screen repository. I don't know if that could be a place where we could put it on the wiki of that. Otherwise, we'd need another one. I think you're right, because it's quite general. And historically, we started repositories based on series, and this is a bit beyond that.
Yeah, what if, because what other molecules to screen, that's an overarching theme. What other techniques / data we need? So then you could put modeling there, but you could maybe also say if we need some, like for some compounds, we might want some mammalian cells done. So some toxicity screening, not only in larvae, but also in something else. We could also put how we do toxicity screening, because that's a technique, because for me, it was also difficult to put that somewhere.
Yeah, we need like a title that's like everything generally, but things that need to be done or need to be that we're interested in. But we have all these repositories existing already.
Because many of these things are overarching and would be relevant for the K-toxamides or the Phenomals, but probably also for Series 5 as for Series 3.
Yeah, absolutely.
So these things which are overarching, it would be good to have a repository, but we have to think of a good title.
Have, just the general start here repository, which logically would be the first repository that people look at if they are new to the page. So we could try that.
Could we also make that a little bit more order, because now it's in the middle? If we put a 0.1 before it, would it then be on top?
You can't change a repository name. You should, I believe, I don't know, Matt, if you know any more about this than me, they should logically be up the top the more recently they've been updated.
Okay.
Yeah.
So
yeah, and it's not an issue that does that. I think you have to change like the front page README and stuff, and then it bumps up.
Because we've also, these two up the top, Series 1 and Series 2, have been pinned. So we could pin the general one at the top and move these two.
Yeah, maybe that's better, because the general one is the one people should spot on right on.
So shall we try that for now? Pin the general one and use that as our general repository.
So we in the what other molecules to screen thing, we have an issue open about the kinome, and I guess that raises the issue of where Mickey's results are. Have they gone some? Because that's a general approach in the way that you might think is similar to the informatics approach that we're doing about trying to find molecules that could be screened productively, by similarity of protein. I don't know. It's a tricky one. You know, we do need something which is overarching, but that general start here was kind of meant to be for very general thing. I mean, it can be used for that if it's empty. You know, we're not using it. We can use it for that. But it's meant to be a place where people there new would go along. If we start having a complicated discussion about bioinformatics, I don't know if people are going to go for that.
Should we maybe try pinning, like, obviously we don't want to complicate things too much, but like keep the general one extremely general and pin as well the what molecules to screen up the top, and have it clear in the README somewhere that it's not just molecules.
Yeah, we can change the README.
Yeah, we can't change the title, but if we say, you know, if you're interested in modeling, etc., as well, go to this one.
Yeah.
Yeah, because modeling could lead to what other molecules to screen.
Yeah.
Yeah.
Okay.
Right.
And I might have a go at trying to organize that, and then hopefully next meeting I can, you know, we can see if it looks like it makes sense.
Okay, sounds good. All right, moving on to series 2. So so there's some discussion on the mechanism of action for literature search for mechanism of action for the series 2 aminothiazoles. I think Wendy has also created a page to communicate this. Wendy, do you want to talk a bit about this?
So, yeah, this is, so this is about synergy. Synergy testing is quite easily in the lab to do. So we can make a checkerboard essay when we need to know these kinds of things. Abafungin, I don't think it was active, but I have to double check if it was also part of the pathogen, the global health priority box. So we have to double check if it was in the MMV box. I didn't create all of this. I think Erica did.
Yeah.
And Erica's not here, is she?
No, I don't think so.
Those students who are maybe working on this have been working on it in the last month or two. So I think we should just make sure that Erica tries to give us an update next time.
Okay. Also, in terms of the just literature on MOA and for the series in general, I've done a little bit of like just literature searches when I was writing my introduction for my thesis as well. So it has a little bit, but it has all of 6 series. So I was wondering, like, if I want to put that on GitHub, I can probably just like, here's what Hong's thesis found. It may or may not be of use. So I don't know. Is that something that I can just put on at some place? Maybe the general place that is being created by Clem in the near future?
I think there is somewhere where all the theses are deposited. So Wilson's thesis is also deposited here somewhere. And Mass, we will upload that as well.
I think if you've got suggestions from literature about mechanism of action, it'd be great if you could put those in the relevant repositories. I mean, just put the papers that you think are relevant.
Okay, yeah.
But split them up into the different repositories, I think, because we're talking about different targets.
Okay, yeah, sounds good. Last, the fourth point, which is securing samples for one interesting paper from 2022. I think we're just looking for any updates on this? Does anyone know if Louisa Cruz, Dr. Cruz, is still on this?
I think that is a bunch of emails I occasionally get, which is the subject of an MTA between Erasmus MC and DNDi, right? Isn't that what that's about?
Yes, so they need an MTA with Erasmus MC to be able to send those, and it finally, because it was about some wording here and there, so it finally now complete, signed. So yeah, that happened last month, so it's now signed.
We just need Louisa to send the molecules. Is that the next?
Yes, so the MTA is in place now, so now she can send.
Okay, cool, great. I'll make sure that's on the action list.
All right. Awesome. Next one is series 3 from phenothiazines. First item is Dr. Rainer to update on any of the phenothiazines that have been sent through and just progress of students. Thank you, Dr. Rainer here as well.
Hello, can you hear me?
Yes, we can.
Oh, can you still hear me? Good. I'm in a log cabin in the woods, so the internet's not great. We sent probably about 20 series 3 compounds through that we finished off making and purifying last term. So far on the ones that we sent, I don't think anything's very good. So we'll see on these ones coming through. Yeah. So we've got procedures down really well now for making these phenothiazines. So they're really quick to make, but whether they actually are going to do anything, we're going to focus this coming academic year on changing the core and keeping the chain standard across them, and we'll see what we get through that way. But yeah, we sent some through. We'll see if they come to anything.
Yes, so they are on the list to be tested. So I don't think, I think by the end of, well, in the next meeting, we will have some of the preliminary results.
Yeah, great. Yeah. We've probably got 15 or 20 students working with us for the academic year. So we'll have a think of some compounds to make and post them when we
because what we were thinking was we're going to keep the tail of the Curaxin compounds. So the ones that Lori sent that were potent, we're going to keep that tail and just change the tricyclic head. See if we can optimize it.
Okay, so yeah, the next point is testing at higher concentration. I guess that will be also updated next time. Is that right, Wendy?
So the concentration we tested are the standard concentration. I don't think going in vitro higher, that doesn't make sense because that we can't reach that in vivo anyway.
Okay. Okay.
Don't know how that got on the list. Higher doses because there's got to be a cutoff.
So does that mean that we or when do you think that it will not make up, like we just will be doing high concentration, any higher, that is?
No, what we found out, we screened recently also a 6,000 compound library.
Yeah.
We're still verifying that, but it was actually better to screen lower concentrations than higher concentrations, because with the high concentration you get many hits, which in the end you cannot reach in vivo. So if you can't reach it in vivo, then there's no point in screening further, of course. So that means that 125 is already relatively high. We could also consider to go lower than that, but on the other hand, I also find the beauty of having all these compounds screened at fixed concentrations that you can also compare maybe a little bit easier. So yeah, that's why we stayed for now as it is. And sometimes we simply, if we have to screen a lot of compounds, add a lower concentration as well to narrow the workload down.
Yeah, okay. That makes sense. Thanks, Wendy.
Now, the curaxin compounds that were going to be screened in the larvae, those were not sent, right? Lori has stock issue shortage.
So all the compounds which arrived are currently in the list. And if something was sent and it's not arrived, but the shipment date is also not in the file, yeah, then I simply don't know.
Yeah, then someone will have to follow up with Lori to see if we can get any more of those curaxins.
Okay, there's one question from Matt for Peter about data on the synthesis, so NMR data, etc.
So yeah, we have it all, and it's all in UK Lab notebooks, I think it's called. We can just upload the link, and I'll stick that in one of the open issues or something, right where it is. But yeah, all the data is available.
Sounds good. Thanks, Peter. Next point is Series 3 wiki is now up and running, but I can also see that there are some like unfilled in section as well. So is this still ongoing work, or is this already kind of like a stage where we're happy? Does it need to be assigned to anyone?
Just kind of, but yeah. Well, what happened was we're kind of pivoting from the chlorpromazine, phenothiazine type compounds to more of the ones that are similar to the curaxins. So it's kind of a little bit in flux. Kind of going more towards the curaxin ones because those tested all right. Well, I don't think Lori has the original parent molecule, which is curaxin 137. Bernard, do you have any of that, or because I know it's used in cancer research, so if anyone has any stock of it, could be worth just screening the parent molecule, which is curaxin 137.
Owen, you are muted.
Oh, sorry.
Okay.
So next one is just a comment. So the tails of the series seems to be fixed and the head, but the head seems to be able to be optimized. Is any?
Yeah, that was what I gleaned from reading the paper that Lori's group published on the curaxins for the trypanosomes. So they were able to modify the carbazole head, the tricyclic head, but anytime they changed any part of the tail, it lost activity, as far as I can tell. Other than that, there was one compound where they had a pyridine or morpholine as a replacement for the isopropylamine, but anytime you deleted that basic amine on the tail, it didn't work. You couldn't replace it with a sulfur or oxygen or anything.
Yeah. And has this been captured in the wiki as well? Did someone add it into the wiki for Series 3, or is this just here because we can just move that over there?
We don't have SART.
Go ahead.
think anyone's made a SAR table yet. Peter, you said you were getting students to look at that, right?
Yeah, they're on way with that at the moment. There is not a lot to, but we'll get a visual update on that, and we'll pop it on the wiki.
I will just leave that to Peters for update.
So what I just mentioned is that we do have an additional library where we screened 6,000 compounds, but we didn't put it up yet because it was, we simply need to verify a few things here and there. So that will take still some time to go because Ma is verifying these things. But we will put it up as soon as we're confident with all the data. That will give us new leads maybe as well, but also all these questions about compounds which were developed for something else. We can simply look there up if they were screened and what the results were. The only thing is these were screened at much lower concentrations because they were 6,000 compounds and we needed to narrow down the hits. So these were not screened at 100 micromolar or 25, they were screened at 2. So that's really lower.
Yeah. Thanks, Wendy. And the last point on here is there's 5 of the correction compound screen for toxicity that is very non-toxic. And I think the question was what MyOS codes have we added into the have we added this into the master list yet? Provided MyOS codes for these.
So I put in the action, can anyone clarify which compounds were tested? I answered that with the MyOS codes you see and the...
Okay, so those are the 5, essentially. So that point has been completed?
Yeah.
Okay. And that data has been added to spreadsheet. Okay. Awesome.
So I guess it's just a matter of following up with Lori then about getting whatever she has left in stock.
Yeah, but with the point that it's already almost September, so if things arrive late, then they are too late for this larvae season and have to wait till next larvae season.
Oh, what are the start and end dates for larvae?
So larvae, we usually start somewhere March, April and end October. If we are lucky, we can do something in November, but it depends on the weather.
Okay, thanks, Manny and Anthony. Is there any more points for series 3 before I move to series 5?
No, but I didn't hear. But Bernard, do you have T-Rex 137 in stock, or? I know it's used in cancer research. I was just curious about that.
Yeah, I am not too sure about that. Does anyone have more information on this?
Bernard, I think that was a question from you, from Anthony.
Don't remember anymore, but maybe I've something in mind that the Cleveland Clinic has made some research on that compound, but I'm not sure. Can't say.
Okay, because it would be interesting to test the actual parent molecule, because if I'm not mistaken, the batch that Lori sent did not have that original molecule. It had an amide analog when the parent compound has acetyl groups. It had acetamide instead of the acetyl groups.
Yeah, I think the amides were not so active in some previous papers about that compound class. So I think it's better to go on with the synthesis pathway Peter has started.
Yeah, I think it might not even be worth spending the money to buy the parent compound, because it might be that one of the compounds that we proposed where we hop the tricyclic might be better. All right, that makes complete sense. All right.
All right, let's move on to series 5 then. So I see 2 more items for Mars. I guess we'll just leave this for next meeting. Is that okay, Wendy? Mars is drafting results for series 5 into a paper, and also
Yes, she was in contact with Andrea. They did do a SAR, and she's drafting everything up. So let's leave it for next month.
Okay. And next one, there is some volunteers needed for master list to do renaming benzimidazoles. Have we got volunteers for this yet?
I don't think I can see from the structure myself what it is, because I can't read those smileys. So I think that's somebody from the chemist team who needs to do that.
We could ask Andrea if she could find a student maybe to help with doing some of that.
Yes, okay, so probably we'll
Yeah, Clementine says that's a good idea.
Yeah, because I don't think she knows the most about which ones are which, and she has a list of which ones are benzimidazole already by code.
Okay, let's test this and ask Andrea. And are there any other labs with related compounds that we should contact this? Do we know any more labs?
I think that's also a question for Andrea.
One more for Andrea.
I know she has like a colleague at a different university. I want to say Imperial College in.
Yes, it was ICL in
Yeah.
Charlotte. Don't remember the surname, but she should be also have an account at GitHub. And she was also involved in that DNDI project originally.
Yeah, that's also what she mentioned during the discussions with Ma on the SAR, that this was all from a DNDI project originally.
Yes, where they're looking at something else. Yeah. And that note, I think, has been in the agenda for like a long time. Is it like, are there specific compounds that we want with the SAR? Does anyone know? Or is this we need to ask Andrea?
Yeah, it's fishing, I think, around the same core.
Okay.
Yeah, I think there was one compound, the hits were having a chlorine and a bromide somewhere in the ring at specific places. And we had 2 chlorines, we had 2 bromides and 1 chlorine and 1 bromide. But the other way around, we didn't have.
Okay.
Something like that it was. I'm not a chemist, but I hope you understand what I mean.
Yeah. So we maybe should see if there's anything particular that we want, and we can put on a list somewhere of this, and this would be good to test, rather than just any benzimidazole.
Yeah, because the core structure of these molecules for the rest was exactly the same.
Okay.
All right. Next up is Ketoxine Series 6. Dimitri not here today. Matt, do you know if Dimitri has done this or update on these two? So updates on SAR for Ketoxine and share priority list for further in vivo testing.
Yeah, sure. That it'll be coming. Yeah, he submitted his PhD same time as you, Hong. Congratulations, by the way.
Yay.
And so there's diagrams there ready to go, and we're thinking about changing them a little bit for a paper. So as those come ready, they will come online. Both things that were mentioned then, so the SAR diagrams and rationale for testing any other compounds in vivo, given the results that we've seen to date, but also the DMPK data that we got from Dundee, just summarizing that for Series 1 and Series 6, and making sure that we've all seen those data as well, because there are a couple of interesting things that we might want to think about.
Okay, so that will be coming soon from Dimitri.
Yeah.
Okay, sounds good. Okay, last section on other items. So first one is for Wendy. So this is about collaborating with Melissa on sporotrichosis. Do you have any updates on this, Wendy?
No, not updates. I will be visiting her next year as well.
Okay.
But yeah, we still needed to find time to create this library and ship it. So we do have all the compounds. It's simply finding time to fill it in a 96 well plate in a normal order.
Yep. Okay, so this one is coming up item. Next one is information about niclosamide. Is it related to the sporotrichosis, or is this a separate item? Okay.
Paper was just published, Bernard said.
Yes. Yes. I made a comment today and provided a link to the open access paper. It was published late in July, and yeah, everything's fine about it.
And QID is collecting information about off-label drug use, and do we still need a pharmaceutical contact, maybe for the NDI? Is this something that we're still working on?
Pharma contact through DNDi. I'm still trying to find a time where I can talk to Louisa briefly. We keep missing each other. And I'm going to ask that question when we finally talk, so that can stay there. The QID thing, I'm not sure.
Yeah, I think the QID is not for us. It was more to mention to you that they are developing or that they are gathering clinical data on the current drugs available. So terbinafine, posaconazole, itraconazole. It's nice that that's been collected, but that will not help us developing novel molecules because those are simply the azoles and maybe terbinafine.
Okay. And 2 action items done. Last item, DFK. Okay, so that's gonna be with Dimitri's upcoming updates as well. So next one is on the global health priority box. Do we have any updates from on analogs from GSK?
So this is this compound that changed the morphology of the grains, right? So yeah, I have had no reply from GSK. The molecule itself is not commercially available other than as a synthesis on demand for a large amount of money. No analogs seem available. It really seems to be a one-off. I'm in touch with a company about whether or not we can get it made for us so that we can check it and so that we can think about what chemistry we might use to explore it. But I can't provide an update yet.
Okay. Do we have a structure for this somewhere?
Yeah, it should be linked on one of those links. It should take you back to it. It's very medchemy compound, and it would be a 3 or 4 or 5 step synthesis.
Okay.
Not totally trivial. So it might be quicker to try and get it made somehow, because we don't have a lot of chemistry resource right now.
Yeah. Is that the one of these blue link here? Like,
yeah, I think so. One of those.
Yeah, it's one of the links on the meetings section. I posted the image. It's here. In the monthly Zoom meetings repo, it's MMV.
180-4559.
Yeah, 4559.
Okay, so that's the one that we're interested in. Okay.
Yeah, like a 5-step synthesis.
Okay, so for this compound, so Matt just said we'll just try see if we can buy it, essentially. Okay. And third item is okay. That explains why the compound is desirable.
Yeah, all of that stuff is related to the same thing.
Okay, so that's all. Just pending more updates from you, Matt. Sounds good. Last one is kinase inhibitor library.
Yeah, so Mickey, maybe you can simply update.
Yeah.
For the kinase inhibitory library, we finalized the manuscript and it has been sent out for review. So at Helion, I believe. So I don't think we have any more updates on that topic.
Now, maybe good to mention one more time, we stumbled upon the cell wall integrity pathway there with a few kinases active in that pathway, both in vitro as in the larvae model. We use transcriptomic data for that. We, of course, because these were fungal specific kinases, those there were no fungal specific kinase inhibitors, of course, in the key CGS library. Not much known in literature about those kinases itself. There were a few molecules which potentially could inhibit, but one of them we tested in the larvae was toxic, very efficient way to kill the larvae. But there we were stuck that we know which genes they are, what type of protein they encode, but not what kind of molecules we could try to verify if it would indeed be a good target. The new postdoc, who start next month, will work towards making targeted knockouts in Madurella mycetomatis. That's a technology which currently still doesn't exist, so we cannot verify it by making simply a knockout at the moment. So we hope that in the future we can find, if we see a target like this, can also verify it by making knockouts if it's indeed a good drug target. That would help the field and us in the MycetOS community a lot, I think.
Do we, I mean, it would be great to know if the molecules are on target. But we don't know anyone who is, routinely playing with these fungal proteins, right? Recombinantly.
No, because there was actually not much known about these targets in literature at all when we wrote the discussion. So, Mickey, there was something in Aspergillus and Candida, but not very much, was it?
If I recall correctly, not much was known. As you mentioned, there was something written about Aspergillus and Candida, but that's all about it.
And that's only 1 or 2 papers. It's not like there were 20 or 30 papers on this pathway in Aspergillus. It was only once or twice.
No, it's very limited indeed.
But on the other hand,
really exciting, right? Because these are new targets that we know are going to be important. So it's really great.
And we could also reuse the data we have. So that was also part of this kinase paper. We actually reused all the in vitro and in vivo transcriptomic data, we have to answer different questions. So like with the kinases, we could bioinformatically look if some of them were fungal specific, if they were shared between human, and comparing with Aspergillus knockout libraries. So if there are other libraries which would be interesting, we could do a similar approach of that, of course, as well. So if it's not only kinases, but something else, other types of enzymes, we could reuse the data because it's simply there. So that's maybe also for some people to think of if they have a good suggestion. Maybe Dunja can look into the data we have and take it from there.
Do we have predicted structures of the, I guess we do, right? Predicted structures of the fungal protein versus the human ortholog.
Yeah, so they did this in a project. It was using our data, but they did, they made all those protein predictions. They are online, so you can easily find them. They're also there for human, of course.
And yeah, so all of the data are also already been, I say, the action to link data which has been done. So most of this information are already on the master list. Is that right, Wendy? Or in the manuscript as well? Okay.
Yeah, and we added all the raw data. Well, not the transcriptomic data. That's of course it is deposited, but it's not in the master list because that's too much data. But we added all the screenings we did on the key CGS library in the spreadsheet under a separate tab because the structure of the library was different.
Yes, I think that is all items from the agenda today. Is there, and we have about 2-ish minutes. Is there anybody who have any other business or any questions? All right, sounds good. Thank you everyone for joining the meeting today. Next week. Yes, sorry.
No, nothing. Sorry. I was going to say it was nice talking to you all. But I yawned a little. It's a little early.
It's late for me, so I'm gonna go to bed in about 1 hour or so. It's also nice to see everyone as well. Yeah, I've been kind of just not do anything after I submit my thesis, but I'm slowly coming back to fix all of the stuff that eventually will come back to me. So yeah, nice to see everyone again. Yeah, next meeting. Yes. So
nice
next
to see everybody.
Yeah. So next meeting is that gonna be on 24th of September. That will be our next meeting. So thanks everyone for coming. Yeah,
and remember, next meeting is the odd month, so at the earlier time from this compared to this meeting.
All right, that's good. All right, so let's just end the meeting here, and we'll see everyone again next month.
Bye.
It was nice talking to everyone. Bye.
Bye.